Hi everyone, my name is Sarah and I'm a postdoc working at the European Cancer Stem Cell Research Institute at Cardiff University. And today I'll be presenting to you my poster with the title Wnt regulates metastatic cholangiocarcinoma. So firstly, an introduction, cholangiocarcinoma is a heterogeneous malignancy arising in the epithelial cells that line the bile ducts within the liver. Classically, it's described as a rare disease. However, in recent decades, both incidence and mortality rates are increasing, causing a global issue. Cholangiocarcinoma is described as being clinically silent. This is because most patients present with advanced disease in the clinic, where they may have both local or distant metastasis in the liver, lung, and peritoneum. As such, unfortunately, there are no treatment options available for this subset of patients, with palliative care being administered. Even in patients where cholangiocarcinoma is diagnosed at an early stage, treatment options are very limited. They are offered surgery, resection of the liver. However, a high proportion of these patients do indeed go on to relapse with resurgence of the disease. This is due to the highly invasive nature of cholangiocarcinoma. As such, there's an urgent need to discover novel mechanisms driving the invasive nature of this disease in the hope of developing novel therapeutic strategies for metastatic cholangiocarcinoma. So this brings me on to my hypothesis. Our hypothesis is that Wnt regulates several stages of the metastatic cascade in cholangiocarcinoma, particularly through the Wnt receptor Frizzled-7. It does this by regulating cholangiocarcinoma cell migration, controlling the interplay of cholangiocarcinoma cells at the secondary metastatic niches, such as the liver and lung, and thus by targeting the Wnt signaling pathway at the level of the receptor ligand interaction will be a novel therapeutic strategy for metastatic cholangiocarcinoma. Now, this hypothesis is based on the rationale that Wnt signaling has been shown in the literature to regulate several hallmarks of the metastatic cascade. Firstly, Wnt signaling has been shown to regulate tumour cell migration from the primary tumour. It's also been shown to regulate epithelial to mesenchymal transition, and also the reactivation of cancer stem cells at secondary sites, setting up secondary metastatic deposits, all key events in metastasis. Now, our research lab are particularly interested in the role Frizzled-7 plays in solid cancer. We've previously showed that Frizzled-7 regulates the initiation and progression of gastric cancer. We've also seen that Frizzled-7 does indeed play a role in metastatic prostate cancer. Data by Luke Bolter has also shown that Frizzled-7 does play a role in the primary cholangiocarcinoma. However, we want to investigate Frizzled-7's role in metastatic cholangiocarcinoma. So this brings me on to the results section. So in figure 1A, what we did here is we targeted Frizzled-7 using shRNA, and we were successful at knocking down Frizzled-7 alongside Wnt target genes such as c-Myc, cyclin D1, and TCF1/7. In synergy with this, we also saw a knockdown of the ability of the cells to migrate, whereas when we overexpressed Frizzled-7, there was a significant increase in the ability of the cells to move. Not only are we targeting Frizzled-7 specifically, but we also use a drug currently in clinical trials called LGK974. LGK974 is a potent inhibitor of the porcupine enzyme, which is responsible for the secretion of Wnt ligands. And what we saw in figure 2A is that in both intrahepatic cell lines, so CCLP1s and SNU 1079s, we were able to successfully inhibit the cell migration. This pattern was also seen in the extrahepatic cell line TFK1s, where we also saw a significant reduction in the ability of the cells to move. When we specifically looked at LP1 cells treated with LGK974, we were able to see that this drug could significantly knock down the gene expression of MYC, cyclin D1, and ROC1. More interestingly, we could see that this drug could also inhibit the expression of CD90 and CD44. And in the literature, these genes are shown to be involved in drug resistance and are cancer stem cell markers of cholangiocarcinoma. This brought us on to carry out a colony forming assay, which is shown in figure 2C. What we did here is we seeded cells at a low density in 96 well plates in several different conditions. So we coated the cells with matrigel or liver or lung ECM to recapitulate the metastatic niche. And what we saw here is that LGK974 was successful at inhibiting the ability of the cells to form colonies both on plastic, matrigel, and more importantly, the lung and liver ECM, suggesting that the inhibition of Wnt ligands does indeed inhibit the ability of the cells to form colonies alongside stopping their cell migration. To further recapitulate the peritoneal metastatic niche, this brings us on to figure 3. We in the lab have MET5A cells, which are a normal mesothelial cell line. We take the conditioned media from these cells with and without LGK974. So we have conditioned media with and without Wnt ligands. And what we see in figure 3 is that these cells are more likely to move to the peritoneal microenvironment when Wnt ligands are present, because as you remove the Wnt ligands, there is a dramatic decrease in the ability of the cells to move in both LP1 cells and TFK1 cells, suggesting that Wnt signaling from the peritoneal metastatic niche does indeed promote cholangiocarcinoma cell migration. Now, these key in vitro experiments led us to carry out an in vitro experiment, which is shown in figure 4. What we did here is we injected 10 female nude mice with LP1 cells in the peritoneal area. These mice were treated from day 0 with either DMSO in corn oil or LGK974 in corn oil from day 0, 5 days a week for up to 5 weeks. And what we saw at the end time point is that there was a significant reduction in the number of peritoneal metastases in the drug-treated mice. Alongside this, we also saw less tumours attached to the liver, which is depicted by the graph showing the liver weight. Moving on, we also wanted to see if there was any microscopic lesions within the liver. And what we see here in figure 4B is that by using the human anti-mitochondrial marker, we were able to depict metastatic deposits within the liver, which was confirmed by using the proliferation marker Ki67. What was interesting is that in the drug-treated mice, there was no metastatic deposits found, suggesting that this drug can significantly reduce both the peritoneal and liver metastatic deposits in vivo. So to conclude these key in vitro and in vivo data, this suggests that targeting the Wnt signaling pathway at the level of the receptor ligand interaction can successfully inhibit cell migration in cholangiocarcinoma cells and also colony formation of these cells at secondary metastatic niches. What's more is that we saw in vivo that this drug was able to significantly reduce the number of peritoneal and liver metastases. What's more important is that no toxicity was seen in Wnt-regulated organs, such as the stomach and intestines in these mice, suggesting that LGK974 treatment would be therapeutically advantageous for inhibiting cholangiocarcinoma metastasis. We would like to thank AMMF for funding this project. Thank you very much.